Hi everyone, in this lesson we are going to continue looking at the specific immune response, but today's focus is going to be on T cells. So our keyword list looks a bit longer, but some of these we have met before, such as antigens, antibodies, helper T cells and cytokines. A few of them are new, so remembering we're just adding to the list that we've already got, this shouldn't be too daunting. The learning objectives, hopefully you'll be familiar with these. We are going to be focusing principally on describing the cell-mediated immune response and getting those keywords on the previous page into that response answer. All right, so just a recap of the immune response. Remember that even though we're talking about them separately, both responses happen at the same time. They depend on each other and they help each other. This lesson is going to be focusing on the cell-mediated response, which is associated with T cells, and there's three different types we're going to be looking at. And these cells are involved when pathogens get inside cells. So that's very different to the antibody or humoral mediated response, where we are inactivating pathogens when they're on the outside. So in this cartoon, we've looked at this before. We're just looking at side A on the left here. We're going to be talking about this process and how it works. Again, just a reminder, the humoral response is also happening concurrently, but we're just not going to focus on that today. Again, this is just a quick reminder of what happens. The bone marrow is where B and T cells are made. However, T cells go to the thymus where they mature before entering the lymphoid tissue. As with the previous lesson, antigen-presenting cells migrate towards the lymphoid tissue and they present these foreign antigens to helper T cells. Helper T cells, we know, secrete a chemical, a cluster of chemicals called cytokines in response to this antigen-presenting cell, and that initiates both humoral and cellular responses. The next type of T cell that we're going to be looking at are called cytotoxic T cells. Cytotoxic T cells are sometimes known as killer T cells or killer T lymphocytes, and they work to destroy antigens. So another point of contrast between this and the humoral response is that these type of antigens are most effective against bacteria and viruses inside the cell. This short video you can watch at your own pace. I would recommend watching that before you move on to the next section. So before we get started looking at the cell-mediated response, let's look at the different types of T cell and some facts about them. So we know that T cells are matured in the thymus, and we know that T cells work intracellularly. And there are a few types that we need to look at. The first one we've met before, the helper T cells. These activate both B and T lymphocytes by secreting chemicals called cytokines. The next one that we haven't met before is the killer T cell or cytotoxic T cell. These are more aggressive than the antibodies produced by the B cells because they destroy target cells by secreting a chemical called perforin, which we will talk about in a moment. The T cells are also involved in transplant rejection, so they provide resistance to any foreign cells entering the body. Unfortunately, that also includes transplanted organs such as kidneys and so on. The third type of cell that we're going to be focusing on are the suppressor T cells. So once your immune response has cleared up any pathogen that's in the body, you need to turn off or turn down that immune reaction. And this is essential because you don't want to be having an immune response when there's no pathogen present. That could lead to inflammation or allergic reactions as outlined in point 3. All right, so this diagram here is a really good summary of what's going on from start to end. So just to recap, our macrophages will ingest or phagocytize a foreign pathogen. It will digest using lysosomes, and it will present some of these antigens on its cell surface. So this macrophage becomes an antigen presenting cell. This will migrate to the lymphoid tissue where it will present these antigens to helper T cells. The helper T cells will stimulate B and T cells, but chiefly in this picture we're looking at T cells, or killer T cells in this case, which are mobilized to destroy the antigens which are inside our cells. So thus it destroys our cells as well. We talked about the other two cells, so we're just going to go down to this last category here, the memory cells. So we know that there is immunological memory formed from B cell activation, and the same thing happens with T cell activation, and we'll talk about that at the end of this presentation. So just a reminder of what viruses actually are. They're very simple and they're non-living. They are simply a nucleic acid wrapped around in a protein coat. So therefore they require a host cell in order to replicate. They need to take over the host cell organelles so that they can produce new proteins and assemble those new parts. So now we're going to be looking at the cell-mediated response. I provided you with two types of cartoons that outline that process, and we're just going to go through this step by step, but you can choose which one helps you to understand that the best. Initially, as I've talked about before, antigen presenting cells present these foreign antigens to helper T cells, and these helper T cells produce cytokines. Cytokines will stimulate activation of B cells, which will then mature, proliferate, become plasma and memory cells, and then become active antibodies. And we'll also have another group of cytokines which stimulate the production of killer or cytotoxic T cells. So in this diagram, we can see that this antigen presenting cell is stimulating a helper T cell. It releases cytokines, and that will do two things. If we have had this pathogen in the body before, memory cells are stimulated, and they will figure out what shape of pathogen that is to make a complementary antibody, or we undergo the initial immune response. Cytotoxic T cells are activated, and so we get two things happening here. We get activated cytotoxic T cells or TC cells, and we also stimulate the production of memory cells. Just like what happened with B cells, they make plasma cells which make antibodies, and at the same time as making plasma cells, they will make memory cells that remember the shape of the pathogenic antigen. All right, so how do T cells work? In this diagram, you can see that our cell, so this is, let's say, a human cell, is infected with a virus. That virus has antigens on its surface that you can see in that diagram. Some of those antigens are presented to the cell surface, and the cytotoxic T cell can actually see that. So what it does is it binds to the antigens and secretes the first chemical, perforins, and what that does is it creates pores. So it punches a hole in the cell membrane. And what that does is that allows liquid to either rush out or rush in, and that lyses the cell. Another type of chemical is secreted, known as lymphotoxins. Now, what they do is they activate the cell's own self-destruct mechanism. The cell will destroy itself. It acts in a sacrificial way to prevent the virus from replicating and wreaking havoc around the body. Now, one thing I do want you to notice, again, another point of difference between T and B cells, is that these killer or cytotoxic T cells have antigens attached to their surface. Remember that B cells produce plasma cells, which then released free antibodies. So that is an important difference. So these memory cells that we talked about up here, what happens to them is they remain in lymphatic tissue and they're stored. So the next time this pathogenic antigen is presented, these cytotoxic T cells will mount a response much faster, much quicker, and with much greater intensity. And that is the basis of immunological memory. So in this image here, I'm just showing you an electron micrograph of a cytotoxic T cell actually lysing a cancer cell. So this is what it looks like in real life. So we're going to be looking at lymphokines now. So a lot of these words sound similar, but let's just go with it and we'll go back and review it in a moment. T cells produce lymphokines, and these are chemicals that attract additional immune cells. Essentially what they're trying to do is mount a more intense response by the immune system. So it stimulates macrophages that will then go ahead and engulf that infected cell and its contents. So it intensifies that phagocytic action. So just as a summary for what's going on here, let's go from the beginning and work our way through it. Pathogen is engulfed by a macrophage through phagocytosis. The macrophage will destroy the pathogen and release fragments of that pathogen by exocytosis. However, some of that pathogen fragment are presented on the cell surface. So this macrophage now becomes an antigen presenting cell. In step 2, the antigen presenting cell will migrate to the lymphatic in the lymphatic system, and it presents that antigen presenting cells. It presents those foreign antigens to helper T cells, which will then chemically stimulate B and T cells to become active. Helper T cells do that by secreting chemicals called cytokines. Cytokines, a group of them, will stimulate B cells, another group of them will stimulate killer T cells. Once these killer T cells are activated, they will migrate towards that pathogen, remembering that they have their antibodies on the surface of the cytotoxic T cell, and they bind with that foreign antigen and release a chemical called perforins. And these make pores in the cell membrane of that infected cell. In response to these perforins being released, the infected cell will also secrete lymphotoxins. So it's going to initiate its own self-destruct mechanism and help get rid of that pathogen. After we've had perforins being released and lymphotoxins being released by the infected cell, the killer T cell will then go and secrete lymphokines. These are chemicals that attract additional macrophages and other cells involved in the immune response to engulf that dead cell and its contents. So this increases the phagocytic action of macrophages. Once that immune response has been carried out, so stages 1 to 4 outline that immune response. The next group of T cells come along, the suppressor T cells, and they will eventually slow down that reaction and stop it when there is no longer a need for cytotoxic T cells and the like. Okay, so this next slide, you have this picture in your textbook and you have it in your booklet as well. Again, just to reiterate what's happening in a slightly different form. Non-self antigens enter the body. They become engulfed by a phagocyte, which becomes an antigen-presenting cell. That migrates to lymphoid tissue, which then presents it to a helper T cell. Helper T cells secrete cytokines, which stimulate the B and T cells. Killer T cells will then leave that lymphoid tissue and seek out the antigen that's causing the issues. It will secrete perforins and destroy it. The cell that is infected will secrete another type of chemical, the lymphotoxins, as you can see here, and that will initiate the cell's own self-destruct mechanism. The third chemical that we talked about were the, the lymphokines. The lymphokines, remember, attract macrophages to increase the intensity of phagocytosis and get rid of that pathogen. After that's all taken place, the suppressor T cell will dampen down that immune response. Okay, hopefully that was pretty straightforward to follow. This is a flow diagram that was from a 4-mark question in the 2019 waste paper. What I would like you to do is pause the video and see if you can identify P, Q, R, and S. Okay, welcome back, girls. Hopefully you've been able to work out what these are. If we've got an antigen here and it presents the antigen, then this must be the antigen presenting cell. Where that goes to is the lymphoid tissue and it presents it to the helper T cell. So you've got to be really specific with which type of T cell you use. If you just said T cell, I'm afraid you don't get any credit for that. When we look at this side, at the humoral response, B cells divide and they produce memory cells, but they also produce the cells that carry out the action. Those are the plasma cells, and these will secrete free antibodies. So that's 1 mark per correct box. Hopefully you got those right. This is a table taken from page 30 in your booklet. It looks quite busy, but what I would like you to do is to take the time to read through each list and look at the points of comparison and similarity. So you could maybe color code that to see which parts are similar and which parts are different. So for example, the maturation both happens in different locations. In B cells, it's the bone marrow, whereas in T cells, it's the thymus. However, their point of similarity is that they're both produced in the same place. There are some key differences here, such as what type of cell is produced. We talk about the B and T cells acting differently because of plasma cells and the killer T cells. However, they are similar in the fact they both produce memory. But again, a point of dissimilarity is where these memory cells live. So in B cells, they spread to all body tissues, whereas T cells remain in the lymphoid tissue until required. So that's something that will take a bit of time to read through, girls, but it's very worthwhile and it's a really good summary of what goes on in both types of immunity. Okay, so we're getting to the end of this lesson now, and we're just going to be looking at immunological memory. So in this cartoon here, you can see that the primary response has got many more stages than the secondary response. The secondary response leapfrogs all this initial stuff because memory cells have been created and we can therefore get to work much faster. So this is a form of adaptive immunity. And this phrase here, clonal expansion, what we're talking about is that process of mitosis where we create lots and lots of these cells, these clonal cells, some of which became memory cells, remember. That means that when we have a secondary exposure to a pathogen of the same type, remember it has to be the same type, then that reaction goes much faster. Memory cells can actually remain in the human body for many, many years, decades in fact, or even a lifetime, depending on the type of memory cell it is. But what we really need to understand about this is a secondary infection, if that occurs, then memory cells will react much faster, more vigorously, and they will take much less time to react to that pathogen. Therefore, we don't normally experience symptoms and we don't normally see any negative consequences as a result. In this picture here, this is a classic primary-secondary response graph. Hopefully, if you've done your pre-reading, you should be able to talk about this graph confidently and realize that the concentration of antibodies is very, very different. One point I'm going to pick out here that I would like you to notice is that initially the antibody concentration is at 0, whereas at this point it never goes down to 0, and that is because of memory cell formation. So when we have a secondary exposure, our response is much faster, it persists in the body for longer, and we have a higher concentration of antibodies. I would really like you guys to be working on page 26 of your booklet to write out a model answer to the primary-secondary response, and we will touch on that when we start talking about vaccines.